biology from Swarthmore College and a PhD in biology from Stanford University. Her group utilizes computational modeling, targeted laboratory testing to study molecular mechanisms underlying adaptive traits in the face of heat, drought, nutrient limitation, and pests. They study a variety of organisms, including model crops, orphan crops, medicinal and desert plants, as well as C. elegans, fungal pathogens, and piezophilic bacteria. So a very broad set of organisms here, which is very exciting. Accompanying Dr. Rhee is Dr. Yanniv Dorone, who is a postdoctoral fellow in her lab. Dr. Dorone received his BS and MS in biology from École Normale Supérieure de Lyon in France. Dr. Dorone also completed his PhD in Dr. Rhee's lab, during which he discovered novel proteins that phase separate into membraneless organelles. And he also collaborated with many labs from several fields, including ecology, neurobiology, and biochemistry, to characterize a water sensor that regulates germination in response to water potential. As a postdoc, Dr. Dorone is exploring more broadly how phase-separated organelles are used by plants to sense their environments, as well as to compartmentalize metabolic reactions. Today, Dr. Rhee and Dr. Dorone are going to talk to us about their work in biophysics, seed physiology, and ecology to discover a water-sensing molecule that controls seed germination. So it is my pleasure to welcome Dr. Sue Rhee and Dr. Yanniv Dorone. Please feel free to start your talk.
Thank you so much, Savannah. Can you see my slide?
Yes, we can see it, and it's in full screen. Perfect.
Okay, wonderful. Thank you for the introduction and the invitation to speak here today. I am super excited to be here and share with you some of the new work from our group, spearheaded by a star graduate student, Yanniv Dorone, who has recently finished his PhD. And he's here, as you've heard from Savannah, to tell you the story through his own words. And what I'll do is to give a few minutes of background, highlighting the problems that drive the research in our lab and a general approach we're taking to uncover the functions of uncharacterized genes. And the first example of which is this putative water potential sensor that controls seed germination. And Yanniv's gonna then take over and tell you the story of this journey to the discovery, and then end with some of the implications this might have on agriculture, as well as plant breeding in the future. So, you know, one of the major motivators of our lab's work today is the projected crop yield loss due to global warming. This figure shows you the predicted loss of wheat yield all over the globe and predicted by a number of different models. And I'm sure, you know, figures similar to this are abound today. And this model is based on only 1 degree Celsius increase and with business as usual scenario. In addition to the projected crop yield loss, it's projected that biomass from plants will become a global primary energy source in the next 30 years. So the bar graph on the left shows you that these are the major sources of primary energy. And today's scenario in 2020 are shown in black bars, and blue bars are the situation that is projected for 2030, and the red bars are the situation projected for 2050. And as you can see, by 2050, biomass will become the biggest source of primary energy compared to the other sources. But a big issue with that is shown on the right, which is that biomass is a big land hog. And so, you know, compared to the other sources of primary energy, biomass takes up a lot of land, and this is going to become more and more of a problem in the future. So I think a big challenge that's facing all of us, especially plant scientists in the next 10 to 20 years, is the problem of how can we get the most out of plants, such as energy, food, and medicine, while minimizing resources and reducing environmental impact. I think that we need to build programmable plants. And in order to do that, we really need to understand very well the major processes that go on in plants, such as development, reproduction, metabolism, and so on, and know how these processes are connected to each other. And so that we could be, so that we are able to manipulate plants as needed. I see a couple of major bottlenecks and problems that prevent us from achieving this sort of high level vision. One huge problem I see is that plant science is hugely underrepresented in STEM. This is data from the Department of Labor from this year. It shows you that the number of soil and plant scientists is just only about, 10% of medical scientists who study human health. And, I'm talking to the choir here, and that, you folks are the future of plant science, and you already subscribe to the fact that plant science is critical because you're getting your PhDs. And part of the reason why I thought it would be really great to have Yanniv give most of the talk today is because I want to show you and not tell you that the future is with people like Yanniv and the organizers of this conference and the rest of you who are getting trained in plant science. The other major problem I see in achieving the goal that I laid out is that there are so many genes in plant genomes, and in fact, any genome that's been sequenced, that are completely unknown for function. These are represented by these black pieces of the pies here. They don't even look like any sequence that's been characterized for function. So we have no idea what they do. And as you can see, they comprise a substantial portion of genomes. And without knowing what these are doing, we are gonna not be able to have this type of programmable plants that we want to build. So this is a problem we've been trying to grapple with for a while. And one of the approaches we've been taking is to try to look at non-sequence based characteristics to predict function. And so we applied that idea to try to look for novel transcriptional regulators in Arabidopsis. So just very briefly, what we did was to take the set of unknown genes in Arabidopsis, group them into families, and then ask which of them have these characteristics that are common in transcriptional regulators, such as high percentages of disorder residues or presence of nuclear localization signal, or the ability to activate transcription in yeast. And we came up with about 43 families represented by 195 genes in Arabidopsis. And then we started characterizing a few of these families, and I'm very excited to tell you that the first 4 families that we started characterizing are taking us into really interesting places. And I think sort of big discoveries and not just incremental stuff, And so today, Yanniv's going to tell you about the discovery of a potential water sensor that's important for seed germination. But we also discovered a family of genes important for controlling organ size by controlling cell cycle and proliferation at the tissue level. And then we also, this is also Yanniv's work, actually, discovered 2 additional families that seem to be important in forming and functioning of stress granules under heat stress, which he's not going to tell you about today. And so I just want to end with this slide that sort of illustrates how I think a lot of bravery is needed to go into projects like this, where there's completely nothing known. You're starting from absolutely scratch, and there's a lot of potential for failures. And I just want to say that Yanniv's bravery really kind of, I guess, succeeded in so many cool ways. And the way he did that was he recruited a really talented postdoc in a neuroscience lab named Stephen Boynams, who unfortunately is not able to join us today. But the 2 of them actually then reached out and built an international team of collaborators, ranging from research assistants to grad students to postdocs to junior and senior faculty. And through this team that they built over a number of 3 or 4 years, they were able to discover so much. And it is my pleasure to now then give up the floor to Yanniv to have him tell you the story himself.
All right, thank you so much, Sue, for the very kind intro. I'm super excited to tell you about, actually, I think I'm sharing the wrong screen, so let me do it again. Super excited to tell you about the project today. Let me share. All right, can you see the screen here? All right.
Yes.
Okay, thank you. So, so today I will be talking to you about a novel seed protein, as you mentioned, that we discovered then that regulate germination during water stress conditions through a process called protein phase separation. And so I will do this in 6 parts today. So first, I will start by walking you through the identification of this protein that we named FLO1. Then I will tell you that FLO1 can form liquid condensates in response to water. I will then show you that FLO1 is involved in regulating germination during conditions of water stress, and that the material properties of the condensates regulate its function. I will then discuss some interesting features about FLO1 splice variants. And finally, I will talk to you about FLO1 homologs throughout the plant kingdom and discuss the potential agricultural applications of this study. So let's start with the discovery of FLO1. So as plant biologists, we're taught that plants have to make 2 particularly important and crucial decisions during their lives. The first is when to flower, and the second is when to germinate. And so at the beginning of my PhD a few years ago, I decided to explore one of them more particularly, and that's germination. And so because the decision to germinate is so important, and because plants only get 1 shot at it, this process needs to be tightly regulated. And so seeds really need to integrate a bunch of different information to make this crucial decision of germinating. So for example, they need to decide if there is enough light, if the temperature is right, if there's enough water, nutrients, or if there are any other factors that would limit its potential, such as salt, pathogen, toxic compounds, etc. And so only once these factors were evaluated and that it appears for the seed that the timing is right, the seed will decide to germinate. And so the complexity of this phenomenon leaves us as scientists with a lot of questions, many of which are yet to be answered. For example, we still fully understand how seeds integrate all the different cues into a binary decision of whether or not to germinate. And this question is actually particularly intriguing in light of the fact that seeds need to integrate this information, all the while having to transition between different physical states. And so here's what I mean by that. So during seed maturation, seeds dry out and they transition into a glassy state. So here I'm showing you the results of a study in pea seeds where local viscosity was followed as seeds dried out. So starting on the right side of the graph here, you can see that seeds start in a fluid state during maturation, and as they dry out, they transition to a viscous state, followed by a glassy state once they're done drying. And this process is actually fully reversible in the sense that when seeds imbibe during germination, their physical state transition in the reverse order. And so because of these properties, the question that we ask is, are prion-like proteins involved in seed biology? And of course, I really need to take a moment to explain to you why we even wondered about that. So I'm assuming that most of you have probably heard of prions, but you may not have necessarily heard of prion-like proteins. So first, prions are just really fascinating proteins that can transmit their misfolded state to normal variants of the same protein. And so you've probably heard about prions when it comes to certain human diseases and other types of animal diseases like the mad cow disease. But prion-like proteins are not prions, but they're called this way because they harbor domains that are similar to those found in prions. And so the reason we got so interested in prion-like proteins is that some of them have these really interesting biophysical features in that they can transition between multiple physical states. So for example, here in green, I'm showing you a prion-like protein that is called fusion sarcoma and that is involved in the human disease ALS. And so it was previously shown that fusion sarcoma can transition between really interesting structures and material states. So from a dissolved state to a liquid state to a gel state and even mature into a solid state. And so going back to seed, it will actually make a lot of sense for some of the proteins, especially those that are involved in regulating seed biology, to have such similar properties. And so it turns out that the seed proteome is actually slightly enriched for prion-like proteins. And so just to give you some context here, we define the seed proteome in a specific context as proteins whose gene expression is very specific to dry seeds. And so now let's look at in the next step as the seed-specific prion-like proteins that came out of our analysis. And there are 14 of those. There are 8 known or predicted transcription factors, 5 RNA binding proteins, 3 of which are actually completely uncharacterized. And then there is 1 protein that was completely unknown. So it didn't have any DNA binding domains or any RNA binding domains. And as Sue just mentioned, in our lab, we're very interested in exploring the unknown and seeing where it leads us. So we decided to focus on this protein that's completely uncharacterized, and we named it FLO1. So the next question that we asked is, what is the subcellular localization of FLO1, and can we get some insights into its physical properties? And so what I will now show you is that this protein phase separates in a hydration-dependent manner. And so I'll define a bit these terms in a minute, but before that, I want to start with a little crash course in seed anatomy. So basically, Arabidopsis seeds contain a miniature plant with preformed embryonic leaves that are called cotyledons, and also an embryonic fruit, which is called the radicle. They themselves are surrounded by a first layer of endosperm, which is surrounded by the seed coat. So in all the images I will show you next, the 2 layers were first dissected out in order to look at what's happening directly inside the embryo. The other part I want to share with you is that you'll see in my presentation images that look like this. So these are confocal images that were taken from wild-type embryos without any fluorescent markers. So we converted the autofluorescence here into a gray scale. And what's interesting is that even in the absence of fluorescent markers, embryos are highly autofluorescent due to the presence of these protein storage, of these structures here that are called protein storage vacuoles. And so when the seeds are dissected in 2 molar salt as a way of maintaining the dry state of the embryo, you can see that we see this highly autofluorescent signal. When we expose the embryos to water, they're a bit less autofluorescent, but there's still the significant signal. And so here's now an image of embryos that this time express FLO1 tag to GFP and that were dissected in water. So just to be clear again, this is a fluorescence image where fluorescence was converted into a grayscale. So what is really interesting is that FLO1 localized to those granules here. So those are the black dots that you see in this image. When the same seeds are actually dissected in glycerin, so as to avoid any exposure to water and to maintain the fully dry state of the embryo, no granules could be observed. So instead, we only see those protein storage vacuoles I just mentioned to you. And so when this exact same embryo that I just showed you in glycerin was washed in water, FLO1 granules were immediately visible again. And so here's the really fascinating part, is that this process is fully reversible. So seeds that were first imbibed in water, but then left to dry out, do not exhibit any granules when they're dissected in glycerin. But if seeds that are exposed to water again after this first cycle of hydration, dehydration, you can see that the granules form again. So what I showed you is that FLO1 forms these reversible granules depending on whether or not they're imbibed in water. But the thing is that right now, these are 2 extremes of a spectrum. On one hand, I showed you embryos that are fully imbibed in water and that exhibit granules in all of their cells. And on the other end of the spectrum, I showed you dry embryos that are not exposed to water and they do not exhibit any visible granules. But obviously, when you're in the wild and in nature, there are different types of water potentials. And so seeds have to to cope with those different osmolytes, different water potentials, etc. So the question is, what's happening at intermediate water availabilities? So to test this, we use osmolytes to obtain intermediate water potentials. And so I already showed you that when the embryo is dry, we don't see any granules. So this is in 2 molar salt. So it is equivalent to remaining in the dry state. When it's in water, so 0 molar salt, you see that there are granules, and there are actually granules in pretty much every cell of the embryo. But at intermediate concentrations of salts at 1 molar salt, we see something intermediate. So some cells do have granules, and some cells have little to no granules at all. And so by quantifying how many cells exhibit condensates under different concentrations of salt, we found that the response to salt is very sensitive and follow this really nice sigmoid curve. So the higher the concentration of salt, the less we see cells that exhibit any visible granules. And actually, just to mention, this is not unique to salt. We observed very similar patterns when we tested different osmolytes, such as mannitol and sorbitol, which indicates that this is a general response to water potential, and it's not just a response to salt. And so the next question is really, what are those granules and what is their biophysical nature? And so what we found is that these condensates are behaving like liquids. So first, by purifying Flow-1, our collaborators at the Sukenik lab at UC Merced showed that it can form spherical condensates when purified in vitro, which is really classical behavior of proteins that are known to form those liquid droplets through this process known as liquid-liquid phase separation. Our closest collaborator on the project, Stephen Boynams from the Gittler lab, used human U2S cells as living test tubes. And as you can see, just like in Arabidopsis, Flow-1 formed these spherical condensates when expressed in those cells. And so additionally, a fluorescence recovery after photobleaching analysis revealed that those granules are behaving like viscous liquids. So all in all, what this shows you is that Flow-1 condensates have liquid-like properties and that they form through liquid-liquid phase separation. So the way to think about it is the same way that you would mix vinegar and oil, and then you'll have droplets within another liquid. This is what we have for Flow-1. So the next question is, what is actually Flow-1 doing at the organismal level? And what I will now show you is that it regulates germination during water stress. So in this slide, I'm showing you the results of a germination experiment on salt conditions. So we have knockout in red and wild type in black. So the red is the Flow-1 knockout mutant. So what you can see is that in wild type seeds, which is again the black curve here, seeds start shutting down germination at about 120 millimolar salt. And so as you go at higher and higher concentrations, less and less seeds decide to germinate, with almost no germination occurring at 240 millimolar salt. When it comes to the Flow-1 mutant seeds, which are represented by the red curve, seeds only start shutting down germination at a slightly higher concentration of salt, which is 140 millimolar salt. And in each of the subsequent salt concentration we tested, a higher fraction of the seed population of the knockout will decide to germinate compared to the wild type. So that tells us that overall Flow-1 mutant seeds germinate better under salt stress. By complementing the knockout mutant with the Flow-1 gene, we're able to rescue the phenotype, and this also provided evidence that Flow-1 was the causative gene. So the different complemented lines here are indicated in blue. And what we observed is that there was a dosage response. So the higher the level of Flow-1 expression, so this is the x-axis, the lower the germination of a given complemented line. So in other words, the level of expression of Flow-1 appears to be negatively regulated, correlated with germination under salt stress. So it may sound great at this point that we identified this mutant that is able to germinate better in salt. But what I want to stress here is that this is a phenotype that's not necessarily beneficial, and it actually can be pretty deleterious if the conditions of stress remain the same after germination. So I'm showing you here 3-week-old plants which were left on different concentrations of salt just after they germinated. So we have 80 millimolar salt on the left at increments of 20 millimolar salt all the way to 240 millimolar salt on the right. So for the non-Arabidopsis people among you, this is how a normal healthy plant is supposed to look like at this stage. But as you can see on salt, all these plants were really struggling for their lives, and unless the conditions change, they will not be able to pass on their genes to the next generation. On the other hand, some seeds decided not to germinate on salt conditions, and they made the best decision because after 15 days of not germinating on salt, when we transfer those seeds to normal conditions, they're able to fully germinate. So this demonstrated that the seeds were still alive, even though they were spending 15 days on salt, and they're basically waiting it out and waiting for better days to come. So the next question that we asked was, why was the condensate state of flow 1 after 15 days of laying dormant on salt? So what's really interesting is that despite having been imbibed for 15 days, flow 1 granules mostly failed to appear. But after transferring the same seeds into normal conditions, so without any salt, condensates were able to fully reappear. So this result, alongside the previous result that I showed you, that after the stress is alleviated, seeds are able to germinate, this suggests that there is a correlation between whether a seed decides to germinate and the condensate state of flow 1. And so, in other words, it appears that flow 1 could be acting as a water sensor here, and that the condensate state of flow 1 acts as a green light to initiate germination. And so now as the next step, I will dig a bit more into the relationship between the material properties of flow 1 and its function. So what we first did is that we dissected flow 1 at the molecular level. And so for the purpose of the talk today, I want you to be aware of 3 different regions inside this protein. So at the N-terminal end of the protein, so on the left side here, we named this first region DS-rich simply because it is enriched in aspartic acid and serine residues. Around the middle of the protein, there is actually the predicted prion-like domain, and this one is enriched in glutamines, prolines, and serine, so we simply named it the QPS-rich domain. And finally, at the C-terminal end, there is an annotated domain of unknown function that is actually really well conserved across the plant kingdom, and I will refer to this domain as DUF. So using this domain delimitation, we set out to dissect the roles of these different regions. So now I'm going to show you the results of confocal microscopy experiments in transient expression assays in tobacco leaves. So as a control, you can see that the wild-type version of flow 1, just like it did in Arabidopsis and in human cells, was able to form granules when expressed in those tobacco cells. However, when we removed the DS-rich domain, flow 1 formed this much larger condensates, which indicates that this domain is important for tuning the material properties of flow 1. And so we were really curious to know why these abnormal condensates actually look like in finer details. So our collaborators at VIB in Belgium performed transmission electron microscopy in human cells that are expressing either the wild-type flow 1 or a DS domain mutant of flow 1. So on the left side here, you can see that the wild-type flow granules appear homogeneous, but on the right side, you can see that the DS mutants had this really interesting structure that look almost like fingerprints, and this is actually indicative of structural order and a more solid-like state. So altogether, this revealed that the DS domain tunes flow 1's material properties, and that when this domain is modified, it can lead to flow 1 forming solid-like structures instead of liquid condensates. So moving on to the QPS domain here, when we removed it, there were no visible condensates that formed in tobacco, and flow 1 appeared as diffusely across the cytoplasm. And so this shows that this domain was really driving phase separation, and this is really consistent with the literature on prion-like proteins, where their prion-like domains are able to fine-tune their material states. And finally, what we did is that we expressed flow 1 without the DUF domain, and we observed normal-looking granules. So that showed that the DUF domain is dispensable for tuning the material properties of flow 1. And so these experiments that I showed you in tobacco and human cells were very informative, but the next question is really what is happening inside flow's native environment, which is Arabidopsis embryos. And in other words, we wanted to know how the different mutated versions of flow-1 would impact germination. So the graph you're seeing now is showing you the germination rates of complemented lines with different versions of flow-1 under 210 millimolar salt. So I'm going to walk you through this graph from left to right. So for the first three genotypes, it's basically what you already know because I showed it to you a few slides ago. So we know that under salt stress, the wild type seeds have a lower germination rate than the flow-1 knockout mutant seeds. And we also know that, as I told you before, that when we complement this mutant with the wild type version of flow-1, the phenotype is rescued. But now when we introduce inside this knockout flow-1 without its QPS domain, the phenotype was not rescued and the germination rates were the same as those of the knockout mutants. So this shows you that in addition to being important for condensate formation of flow-1, the QPS domain, which is the prion-like domain, is also important for flow-1's function. When we introduce flow-1 without its DAF domain this time, the phenotype was fully rescued. So what this tells us is that in addition to being dispensable for the formation of the condensate, it also appears that the DAF domain is dispensable for the specific phenotype. And finally, what was really surprising to us was that the introduction of flow-1 without its DS domain, so the one that formed the solid-like aggregates, resulted in seeds that have a much higher germination rate, even much higher than the flow-1 knockout mutants. So at the cellular level, what is really interesting about this DS deletion mutant is that you can see that they lose their reversibility. And so when we dissected glycerin in 2 molar salt or in glycerin, what we see is that despite the embryo still being in a relatively dry state, so both the 2 molar salt and glycerin maintain the dry state of the embryo, we still observe these abnormal condensates. And if you remember what I showed you a few slides ago, this wasn't the case for the wild type version of flow-1, where water is really needed for the formation of granules. So we don't observe any granules when we expose when we just dissect the embryos in glycerin or when we expose them in 2 molar salt, but we only observe them in when exposed to water. So a really interesting hypothesis here is that since the flow and DS deletion mutants are continuously in this condensed state, that they may need less water to reach a condensate threshold that will tell the seeds whether they should germinate. And so to summarize what I just showed you in all these phenotypes and correlate them to the material properties of flow-1, I'm showing you this, this cartoon here. And so what we know is that flow-1 forms liquid condensates in response to water potential, and that this precise phase separation phenomenon acts as some form of a resistor during water stress conditions. Losing its ability to form these liquid condensates when we remove the QPS domain results in loss of function. However, when we remove the DS domain, flow-1 is trapped in a solid-like state, and we get a different phenotype for flow-1 altogether. So what I showed you so far with these mutants is obviously artificial and it's completely lab-made. And so our next question was, are these findings even relevant in nature? And so the absolutely fascinating part was that it appears that nature had already created a DS deletion version of flow-1, and it's doing it through alternative splicing. And here's what I mean by that. So flow-1 actually has 2 isoforms, a full-length isoform, but it also has a second isoform, which is already which has a severe truncation of its DS domain. So so what we found is that basically nature has selected through alternative splicing a form that is already severely truncated in its DS domain. So we obviously then asked if the properties were similar to the DS deletion mutant that we generated. And what we found is that the second splice form behaves the same way as this DS deletion mutant when we express it in tobacco leaves. And so as you can see here, the condensates that were formed by the second isoform are significantly larger than the condensates that were formed by the full-length version on the left. So the other interesting part here is that these larger condensates can even recruit the full-length version. So on the right side of the slide here, we co-express the first isoform, the full-length one tagged to GFP, and the second isoform tagged to RFP in tobacco cells. And as you can see, the large condensates formed by the second isoform is able to recruit the full-length one. So it appears that when the second isoform is also expressed, it can influence the material properties of even the full-length isoform. And so because of these results, the idea that we had is that in the wild, plant population may have selected different levels of expressions of the second isoform as a way of fine-tuning flow one and as a way of regulating germination in response to their environments. So we tested this hypothesis using the 1001 Genomes project. And this is a catalog of genetic variation in Arabidopsis ecotypes that were collected from across the world. So the map I'm showing you here reveals where all these different ecotypes are coming from. So they come from America, Europe, Asia, Africa, the Middle East. And so what we did is that we collaborated with the Moy Lab at Carnegie, and we more specifically used 478 different ecotypes that had reliable transcriptomic data. And the idea was to test if we could find some relationships between the expression of the FLO1 variants with correlations with any published germination phenotypes or with historical climate data. And so when it comes to phenotypes in the graph that I'm showing you that how seed dormancy is correlated with the expression of the two splice forms. So when it comes to the full-length splice form, so that's on the left here, we found that it is positively correlated with dormancy level. So the expression of the first isoform is positively correlated with the level of dormancy. But if you look on the right side of the slide, you can see that this wasn't really the case for the second isoform, which has a truncated DS region. And so this correlation was actually almost inexistent. And if you look at it, it actually almost trends in the opposite direction, even though it's not significant. But these results really make a lot of sense to us because, as I showed you earlier, the DS deletion version resulted in more germination. So you would expect the second isoform to basically be not correlated with dormancy, which would be preventing germination. So when it comes to the climate data, we found actually strong correlations this time for the truncated isoform. So on the left panel here, we found that the expression of the truncated isoform is correlated with precipitation volumes. And on the right panel here, you can see that the expression of the second isoform is negatively correlated with temperature. So these results made a lot of sense to us because we expected the truncated isoform to be a positive regulator of germination. And so it would make sense for seeds to express more of it when there's a lot of precipitation, but less of it when temperatures are too high. And finally, in the last few minutes, what I want to leave you with is that we should be able to apply all of our learnings of FLO1 in Arabidopsis to the entire plant kingdom. And this is because we actually found FLO1 homologs throughout the plant kingdom and even as far back as in algae. So algal species have only one homolog, at least in the species that are well annotated, but flowering plants have multiple homologs, with corn having as many as 9 different ones. And the fascinating part is that we found natural variation of phase separation across the entire plant kingdom. So what we did is that we sampled FLO1 homologs from different plant species and we expressed them tagged to GFP in tobacco. And you can see that the ability to form condensates is actually even present in species that don't have seeds, such as lycophytes, mosses, liverworts, and algae. So most homologs that we tested form granules, and they're actually of different sizes. So some were much smaller and more reminiscent of FLO1 granules, and others were much larger and more reminiscent of the DS deletion granules. The one exception here is actually this homolog from Austrorhynchus that didn't form any granules, but instead, as you can see, it localized to the nucleus. But overall, what it tells us is that the ability to form granules was widely conserved throughout evolution, and it also suggests that the FLO1 family may have played a primitive and likely an important role given this widespread conservation. So our own favorite hypothesis is that since in Arabidopsis, flow-1 is involved in seed water sensing, that maybe flow-1 homologs were also involved in water sensing already in unicellular algae, and that maybe this water sensing mechanism had a role in the conquest of land by plants. And so this hypothesis is not completely outside the realm of possibilities, as there are actually quite a lot of commonalities between seed proteins and proteins that are involved in desiccation and proteins that are involved in drought responses. And so, of course, understanding how seeds sense water and how they make their decision to germinate is really important from an agricultural perspective. So knowing that there are flow homologs that probably play similar roles in crops could be helpful for agricultural applications, particularly when it comes to seeds. So globally, the demand for improved seeds around the world is growing extremely fast, and it is actually driven by the Asia Pacific region that's really ramping up its agricultural production. And so to give you an idea of how fast the market for seeds is growing, this year in 2021, the seeds market is estimated to be worth $63 billion, but in 5 years from now, it is expected to grow by almost 40% and will reach $87 billion just in 5 years from now. And so, as we all know, there is the question of salinity and drought and our need to develop new crops and better crops. So to put numbers on this problem, there's the fact that 45% of agricultural lands suffer from severe drought conditions, and then 52% of the world's population is seriously affected by soil salinity. And there's also the question of water scarcity that is related, and our need to optimize water for usage in agriculture. And as a matter of fact, actually, our global usage of water has been growing at twice the rate of the world's population. So again, really understanding how water sensing occurs in seeds can really play a role here. And finally, there is the question of achieving uniform germination. And so a common technique that is used by seed producers is called priming. And so the process of priming involves partially imbibing seeds to initiate pre-germinative processes and then dehydrating those seeds before they get a chance to germinate. And so by doing that, partially imbibing those seeds, you're able to prime seeds for germination. So that means that the seeds will be ready for germination, and this will result in more uniform and quick germination. But the problem with priming is that we actually don't really know what are the genetic factors behind it. And so everything that is done in priming so far is mostly done empirically. So I would say that understanding the genetic factors behind seed water perception would be a great way to start. And so finally, we would like to acknowledge the many people who have contributed in many ways to this particular project. So as Sue mentioned earlier, the work today had important contributions from collaborators across multiple fields and labs. So I only got to talk about the contribution of some of the collaborators, but there are even more labs that embarked on this journey with us. So overall, we collaborated with the Giffer Lab at Stanford, the Moy Lab at Carnegie, the Sukenik Lab at UC Merced, the Whole House Lab at WashU, the Tegby Lab at Wisconsin-Madison, George Basel at the University of Warwick, and VIB in Belgium. In our own lab, so the Rhee Lab, we had Flavia Bossi, Benjin, and Elena Lazarus, who actually also directly contributed to this project. And I also want to thank all of our other lab members, past and present, that have all contributed intellectually with helpful discussion. We also are really grateful to our amazing campus and all the plant biology labs on Stanford campus and Carnegie campus. And all their lab members have been super generous with reagent sharing and intellectual discussions on this project. Of course, we want to also thank all the people who have been working behind the scenes and have made our lives really easier. So these are the people in charge of the Carnegie facilities, greenhouse, admin, and IT. And we want to acknowledge the generous contributions of multiple donors, of sources of funding that made this work possible. And finally, we want to acknowledge the scientific interest groups we're part of, and especially the IDPSIG, which is a community that was started by postdocs at Stanford and Carnegie, and that hosts seminars and events about phase separation. And as a matter of fact, some of our collaborations for this project started thanks to this scientific interest group. And so with that, Sue and I will be happy to take any questions you may have.
Thank you. Yanniv, for an excellent talk. Yes, please feel free to put questions in the Q&A, and I will read them to the speakers. Okay, we have a question from Sakin Ruski. Question 1, actually 2 questions. Question 1, since these proteins accumulate in PSV, I wonder if they have a role in the end use, quality, and or food allergy sensitivity.
So actually we don't we don't know for a fact that they're in protein storage vacuoles. So basically the protein storage vacuoles are so autofluorescent that it's really hard to tell where FLO1 localizes in the dry state. So it could be that it is in the protein storage vacuole, but so far we believe that it is actually diffused in the cytoplasm, so around the protein storage vacuole. So this hypothesis sounds really interesting, but we actually don't have the answer to that.
Thank you. Our second question from the same participant is, as proteins crystallize in microgravity, how do you see the role of these proteins in sending seeds to space?
This is a really interesting question. And I think that so something I was thinking about recently is that there's actually, in the literature, there are some proteins that are known to form condensates in response to gravity. And so I believe that it could be also through the process of phase separation. So phase separation is really this really interesting phenomenon that actually, it's a binary decision of whether or not you're in the condensate state or in the diffuse state. And so there are actually a lot of environmental factors that really impact this. And so you have all these different environmental signals that can basically impact this binary output, which is either condensate or diffuse state. And there are a lot of environmental factors that are involved in this. So the one that I talked about today is water, but as next steps, we're interested in other types of environmental conditions. So gravity is definitely one that's super interesting, yes.
Great, thank you for that answer. The participant also says thank you. So I have a question from a plant breeding perspective. Could you kind of just summarize what you think the best steps would be in incorporating these concepts into crops in general, or if you've thought about it in a specific crop, how you think it could be incorporated? Yeah,
so something we've been thinking about is that there are some other results that I haven't discussed today, but we believe that FLO1 could be involved in the formation of the glassy state inside seeds. And so obviously there are a lot of interesting applications when it comes to germination and really regulating those crops to cope with conditions of water deprivation. But there's also the even more interesting question that's even more sci-fi, which is the fact that some seeds are considered orthodox and some are recalcitrant. And so that means that a fraction of seeds actually that are known in nature are not able to desiccate and dry out properly. So a lot of the seeds that we use commercially, like the need for having dry seeds is really important because you can store them, you can ship them. But there are other crops that we cannot use as well because of this lack of ability to preserve them in the dry state. So it'd be super interesting if we can apply FLO1 to make some types of recalcitrant crops more resistant to basically being in the desiccated state.
I actually want to append that some of those crops are like cacao and coffee, which are interesting crops, not only economically but also sociologically.
That's a fascinating answer, actually. And also, I can imagine relating back to Banu's talk earlier would be really important for conservation of these different crops long term. That's fascinating. Okay, we have a question from Joe Feehan. Have you looked at intramolecular interactions between the DS and QPS Rhee domains? And is it possible that the DS domain is autoinhibitory?
So I'm trying to think about what would the DS domain is autoinhibitory mean. Possible DS domain is autoinhibitory. I'm not sure. Sue, do you have an answer to that? Okay.
I think what she's talking about is because when you get rid of DS, you have too much aggregation. She's asking, is the DS wild type version of the DS like dampening aggregation so that it's like basically the full length is able to kind of reach that sweet spot of condensation? And I think she's absolutely right. And the domain that Yanniv didn't talk about, actually there's 4 domains, right? The one that looked like a thin line is actually a structured domain that we know is responsible for nucleation of the protein into condensation. And so the paper in bioRxiv actually goes into that a little bit. But yes, there is this intramolecular interactions between the DS and the QPS in opposite directions. So QPS, and then that nucleation domain is important for the liquid droplet formation, but you can do too much. And so then DS is like preventing it from going too much. And somehow nature has figured out a way to get rid of DS, and so that you can get to too much state. We don't know what that means, but I think Yanniv's sort of given some interesting hypotheses. Does that make sense, Yanniv? I think that's what she was getting.
Yes. And also, and actually we didn't discuss it today, but in the paper that's on bioRxiv right now, we actually found that across different species, there's actually there's certain species that have a longer DS domain, but a shorter QPS domain, and vice versa. So if the QPS domain is longer, then the DS domain is smaller. So yes, there are those intragenetic interplays that we think are happening here.
Great, thank you for that answer. And the participant says, yes, that answers their question. Okay, I don't see any more questions. If anyone else would like to ask some questions here, please feel free, but there will also be an opportunity to ask questions in the breakout session, which will come after our next speaker. So with that, thank you, Dr. Rhee, and also thank you, Yanniv, for a wonderful presentation. It was fascinating.
Thank you. Thank you so much.
We have another question, actually. Is there any connection between flow-1 and the seed's ability to germinate over time? For instance, some plants are able to keep viability for a long time, whereas some do not. I think that relates back to what you were talking about with orthodox versus recalcitrant.
Yes.
But, yes.
And also, so in the paper, something I didn't discuss today is that what we also found, so I didn't get into that, is that the knockout mutant seeds are not able to remain viable for as long a period of time. So what we did is that actually had seeds that were left in the lab. They were growing together from both Columbia and the knockout, and they're actually left in the drawer for 3.5 years. And I was lucky enough to add enough biological replicates for both the mutant and for wild type. And when I germinated them 3.5 years later, we really saw almost a third drop in of germination in the knockout compared to the wild type. So yes, we believe that flow-1 has a role as well in seed viability.
Great, thank you. Okay, now we're going to take a short 5-minute break before our next speaker at 12:30. So please join us again then. And thank you again, Dr. Rhee and Dr. Dorone, for a great talk.
Thank you.